The kind of overarching goal of my research is to develop cell therapy for Parkinson's disease. And to do that, we study development of the brain, how these cells form normally. And then we use stem cell cultures to try to form the same type of cell from stem cells, learning from what we learned from our studies on development. And the third part of it is that we reprogram cells. So we take, for example, a skin cell and reprogram it into a neuron that we can then use to transplant to the brain to cure diseases in the brain. My group had a big breakthrough 2 years ago when we managed to show that you could take human fibroblasts, so cells that you can obtain from the skin, and then by delivering a set of genes to these fibroblasts, we could turn them into functional dopamine neurons. What we're working towards right now is to mimic the development as much as we can in the culture dish, to create cells that are as similar as possible to the cells that you normally find in the brain. I think the long-term goals is then to learn how to use the cells that we create in the dish to transplant into brains of patients with disease, individuals, for example, with Parkinson's disease. They lack a specific type of neuron, the dopamine neurons. So in the long term, we hope to take the cells that we create in a dish and then transplant them into the brain of an individual with Parkinson's disease. And that way we can replace the neurons that were lost and then restore the brain so that we cure the disease. My main driving force is curiosity. I want to understand how a cell is formed and what controls the formation of a specific cell type. And then to learn to use these signals and molecules to myself control cells in the culture dish. And then in the long term, my goal is, together with my colleagues in different fields, is of course to be part of the process that cures disease.